Receptor proteins, I just want to elaborate a little bit more because we're going to see these throughout the rest of the semester. These receptor proteins, again, have a space where they will bind to something, and we call that a ligand. So receptor proteins bind to a molecule that we call a ligand. Sorry, my handwriting's a little messy. And again, this is going to initiate some sort of response so that something happens inside the cell. One such type of receptor protein binds a ligand, and we'll have an example here. This is acetylcholine, binds this receptor. This is kind of a three-dimensional drawing of how a protein receptor looks. And these three-dimensional proteins are composed of several subunits. And once the ligand binds, in this case acetylcholine, you'll get a conformational change. This protein will change shape and basically make a hole. And in this case, the hole allows sodium ions to come in to the cell and potassium ions to go out of the cell. But again, it's specific, so it only allows those two ions to flow through, and it has to bind acetylcholine as a ligand. There are competitive ligands that can bind this receptor protein in this example. Another type of receptor protein that exists is G-coupled protein receptors. Now, these don't allow things in and out of the membrane, but instead activate an enzyme that will, once a ligand binds, so here's its perfect fit, it will activate an enzymatic cascade through something known as a G protein. So this is a G protein complex. Once that ligand binds, this becomes activated and basically results in an enzymatic cascade. So those are our two main types of receptor proteins. Those channel proteins, just to elaborate a little bit more, again, as I said, they have several subunits and they form this channel. And how much they open is dependent on what type of molecule they can let through. So again, as we saw in the previous slide, only a sodium and a potassium could go through that channel. And so there are channels for lots of different ions. There are some for calcium, there's some for chloride, it just depends. And some will only let certain molecules through based on their charge. Here you can see an example of this with these channel proteins. This one is called a leak channel. It's always open, and it's allowing these ions to just freely flow through the membrane and in and out. So a leak channel is always open. Another type of channel protein is going to be the gated kind. This gated channel essentially remains closed until a ligand binds. Again, we're still referring to the acetylcholine example on the previous slide. In this case, you can see acetylcholine molecules bind, causes a conformational change of this protein, which then opens up the gates and allows the sodium ions to flow in, which happens to be down their concentration gradient, and they can go inside the cell. This image here on the left is when it is closed and there is no ligand bound to the receptor. Second messengers, again, are that other type of protein, that receptor protein that I talked about. And again, just to show you a figure from your book with details, this is going to be that receptor that when it binds its specific molecule, then it's going to activate a chemical cascade through the G protein. And an example of this is going to be stimulation by epinephrine. So we have cells in our body that have these receptors, and when epinephrine is present, they will bind the receptor and activate the G protein, which is this pink protein attached to this receptor protein. So once that is activated, the G protein, which gets its energy from guanosine triphosphate, or GTP, hence the name G protein, it will then move over or be activated to move over to adenylate cyclase, which is another protein, but it has the ASE, so that is an enzyme. So it's an enzyme. It will activate adenylate cyclase, which will then convert ATP into cyclic AMP. And the cyclic AMP, or CAMP, is known as the second messenger. The second messenger will then result in activating several different kinases. Again, we have the ACE, which means these are enzymes. So it's basically an enzymatic cascade. So several kinases are activated, and it completely depends on which cell you're in, which kinases end up needing to be activated as to what is going to happen. In a lot of cases, the activation of the kinases can result in things like increase of transcription and translation, which basically means, hey, make more proteins, make hormones, make more receptors to go into the membrane, many different things. And this is important for you guys to know and understand, because second messengers are the majority of the way that pharmaceutical drugs are activating. This is their main mechanism of action. They are acting as ligands that basically will compete against the natural ligand in your body and bind these receptors and activate this cascade. So that is how most of our pharmacological drugs work.